Thank you very much, Jeremy, and thank you for the invitation to speak at this meeting. The aims of the research that I'm going to present to you are to try and produce a therapy that is less toxic and improves outcomes for patients. And the way to do that is to gain a greater understanding of the underlying immune mechanism, to develop more specific disease markers, and we're on the road, but with quite a long way to go. You'll be familiar with this list of autoimmune diseases. These are the kidney-related ones, and it was only in 2009 that membranous nephropathy was actually definitively characterized as an autoimmune disease. The pathology is very specific. On biopsy, there is a very coarse granular deposition of IgG and C3 along the glomerular basement membrane, and at EM level there are these characteristic electron-dense deposits in the subepithelial part of the basement membrane that we know are immune complexes. Now, the presentation, just to put a bit of context to this, it's a rare disease, maybe 10 to 12 cases per million population. But it is an important disease. It's the most frequent cause of nephrotic syndrome in adults. So patients are presenting with really high levels of proteinuria, 10 to 20 grams per 24 hours, and that causes lots of knock-on effects in terms of lipids and loss of clotting factors and thrombotic and cardiovascular comorbidity. So it's a disease of middle age. It can occur in children, but only recently have we had definitive proof that it is an autoimmune disease by the discovery of antibodies to phospholipase A2 receptor, accounting for up to 80% of cases, and a thrombospondin protein with a 7A insert, accounting for maybe up to 5% of cases. And interestingly, the dominant subclass is an IgG4 subclass with both of these responses. So just a timeline. We could have classified this in the 1960s, and we knew that it was immune-mediated, but not until here did we know that it was an autoimmune disease with the description of the first antigen and more recently the second antigen. The problem for patients is that the therapy was derived as non-specific toxic immunotherapy, if you like, in the dark ages of our understanding. And so this is non-evidence-based, empirical toxic immunotherapy, which is still being used as the standard immunotherapy. And now that we've moved into the age of enlightenment, as I'd like to say, don't we need much better, more specific, less toxic therapies to treat this problem? Only now, with Mentor, which is a study of rituximab versus CNI, will this be published this year. But it will be based on monitoring the autoantibody, and so we will have, for the first time in an RCT, a description of the value of this biomarker, anti-PLA2R, as a monitor of outcome. So it kicks off in 2009, and just to give you the background to it, two pieces of work, both published in the New England Journal. In 2009, David Salant's group in Boston identified, using very simple technology, the presence by Western blotting using serum, blotted onto glomerular extracts. They could show that in patients with this disease, there was a circulating antibody in the serum. It wasn't present in other disease controls or normal. And when they actually did the mass spec on the bands, they found it was actually a protein that had been cloned in 1994 as the phospholipase A2 receptor, and that's the cartoon structure. While that was going on in the UK and Europe, we were undertaking a gene-wide GWAS, and we already knew that PLA2R was almost certainly a candidate, because this paper, led by Robert Kletter at University College, identified the two genes that seem to account for the genetic susceptibility to this disease. We'd known since the end of the 1970s that there was a very strong HLA link, and in this GWAS it was identified as close to DQA1, and we get genetic confirmation of the biology that PLA2R1 has a genetic signal, although even now we don't know how that genetic signal works through into the disease. But when you actually look at, so the reference point here, if you like, the benign alleles are represented here, and this is the odds ratio. So if you've got the two copies of the pathological alleles for both genes, your odds ratio for getting membranous is over 100. Now you tell me another risk situation where that level of risk is associated with the genetics. That's a really powerful statement. In 2014, the other autoantigen was discovered, again using similar technology, detection of circulating autoantibody in the serum of patients not present in disease or disease controls or other cases. And again, this is a thrombospondin family member. We've recently screened over 1,800 sera from patients with membranous to try and get a clear incidence of the presence of this antibody, and in our hands it's only 1.2%, having screened all of those cases. Interestingly, some of these cases are associated with the presence of a tumour, and here we see staining of the biopsy for the presence of thrombospondin 7A, and it's highly expressed in the tumour that this person had at the same time. And so we think this is a way of actually understanding what's been known for a long time, that some cases of membranous can be carcinoma associated. We think that the tumour is actually expressing the autoantigen. So the question then is, what's special about these two autoantigens? Why do they appear to be causing an immune response? I mean, they are in the right place. They're on the podocyte cell membrane, and it's the podocyte that gets clobbered by the antibodies that induces the heavy proteinuria. We don't quite know whether complement is required for that, but there is some evidence in vitro that the antibody itself can modulate podocyte biology. The transmembrane receptors, they're quite big, they're heavily glycosylated, they have multi-domain disulfide bonded domains. We're only now beginning to discover a bit about the dominant epitope in anti-PLA2R, and at the moment we know little about what the dominant epitope might be in thrombospondin. And I come back to saying that the predominant autoantibody subclass is an IgG4 subclass, which speaks to most immunologists as saying this immune response has been going on for a long time, and probably a long time even before the patient presents. Now, this scenario and understanding of the interrelationship between the immunology and the proteinuria has been borne out over the last 6 or 7 years. I haven't got time to go into the supported data. We still don't have the classic passive transfer of autoantibody into an animal model to induce proteinuria. So that's the classic standard for saying an antibody is pathogenic. We still don't have that evidence. But the clinical association evidence is that if you remove the antibody, the proteinuria goes away. And you can remove the antibody with a range of classic immunosuppressive drugs. Where the antibody doesn't go away, you get continuing active disease. And where you've induced a therapeutic remission and the antibody's gone away, if the antibody comes back, the proteinuria comes back. So the clinical association is fairly tight for this biomarker actually being a good indicator of disease activity. So what can we learn about the major dominant epitope in this antigen? Well, we expressed different lengths of the extracellular domain of PLA2R, and this is then blotted showing their sizes. Now, if you just use a rapid antibody race to PLA2R, the antibody reacts with all of those proteins under denaturing conditions, and even when you've reduced the protein. But the human autoantibodies only recognize the larger fragments. You've lost the epitope under SDS conditions, and you lose it once you start reducing. So it implicates perhaps some special conformation or disulfide linking as being required to maintain the conformation of the epitope. Well, we wanted to find the smallest of those fragments that we could use for trypsinization and identification of the antigen. So we were pleased to see that we could trypsinize the NC3 fragment and still retain Western blotting, still retain activity by the autoantibody. And that's the procedure that we then went on to follow. But testing just by plasmon resonance, looking for binding of our autoantibody to those 4 fragments, we were picking up activity, although with a bit of variation, even with the smallest N-terminal fragment. So that, in a way, fingered perhaps this N-terminal cysteine-rich ricine domain as potentially where the epitope was. And so just taking it through the protocol of trypsinization, isoelectric focusing, fractionation on SDS page and blotting, and then cutting out parallel bands of the small fragment here, and looking at the mass spec, we picked up a range of peptides that were present from several domains within NC3. And interestingly, having synthesized those peptides, 2 of them, and both from this N-terminal domain, 2 of them showed quite significant inhibition in plasmon resonance study. So the larger fragment, at this stage it's a 31mer here, gave up to 80% inhibition when we were using the autoantibody to bind in ELISA-type experiments. Even the small peptide was able to show significant inhibition. And the binding to the peptide, to the 31mer, was of a reasonable affinity, which surprised us. So this is the structure of that 31mer. Now it's actually a 28mer. We've chopped a couple of amino acids off. We've identified that the sequences in red are quite important for optimum binding, and the distance between them is also quite important. If you make it too short or too long, you tend to lose activity. The presence of the disulfide loop is also quite important. This is our best indicator at the moment of the structure. Rather than use the cartoon version, this is what we think the receptor looks like. There's a ring here of ricin, CTLD1, CTLD2, CTLD3, and then going to 4, 5, 6, 7, 8. And the epitope, as far as we can tell, is just sitting up. If you think this is like a rhinoceros, it's just sitting on the back of its neck here. We're trying to get X-ray crystal structure, which will determine the precise conformation of this in vivo. So we'd got this sequence from PLA2R, and when the thrombospondin sequence, the thrombospondin antigen was clearly in the frame, we noticed actually that it had a sequence in the N-terminal domain of the thrombospondin that shared about a third of the amino acids. And interestingly, some of the sequences that we knew were important, disulfide bonds in the same place, and this C-terminal MCK-QA was quite important. And we wondered, well, how common is this sequence motif? I mean, do lots of other proteins have it? And so just doing a simple blast search only picked up other variants of PLA2R, other species variants. And the only protein that we could see had it was thrombospondin 7A. So it's not a statistical proof, but it shows that this is quite a rare sequence. And isn't it interesting that both autoantigens display it in the N-terminal region of their protein, and they're both on the same cell? we thought we'd better model those peptides just to see if there was any evidence that they actually took up a similar shape in solution. And yes, they do take up a similar shape, and you don't get precise overlay, but you could argue actually there might be, if you like, a pathological shape to this sort of peptide. So we'd made by this time a number of monoclonals, and we found one monoclonal seemed to mimic the autoantibody, and it binds to the relevant fragments. It's under reducing conditions you lose the binding. It is only specific on protein to the PLE2R, which was the immunogen, and doesn't recognize the protein of the other autoantigen thrombospondin 7A. But when we tested this antibody on peptide, again by plasmon resonance, we could see that the monoclonal would bind not only the P28mer, but also there's a dose response effect on the T28mer. So here is an antibody, a monoclonal antibody, that seems to be able to recognize both peptides, but it doesn't recognize both proteins. So if somebody has the answer to that, please talk to me later. And we thought, actually, we better just check with our antithrombospondin antibodies. I showed you a list of 10 cases that we picked out. And we tested them under native conditions by slot blot. And we could show that all of those 10 autoantibodies actually recognize both P28mer and the T28mer. And so on peptide, there is cross-reactivity of both autoantibodies, but on protein, there is not. The antibodies appear to maintain specificity for one or the other. And the current dogma is you don't find patients who have both autoantibodies. You're either an anti-PLA2R person or an anti-thrombospondin 7A case. Having screened 1,800, we found 2 cases that show that you can develop both antibodies, but it is a very rare situation. So we're left with the situation here where the autoantibodies are specific on protein, but they cross-react with a common sequence motif at peptide level. And to me as an immunologist, that says actually that's quite interesting, because we know when immune responses kick off, antigens get degraded, peptides get presented. So maybe there is some preference for presenting this particular pathological shape that kicks off the autoantibody response. And so we're very keen to try and follow that up. So how can we exploit this knowledge? Well, we've spent 3 years or so collecting a series of patients through an MRC-funded study where we've collected HLA-typed cells, we've frozen them, we've got different clinical outcomes. And we're currently undertaking an immunoabsorption study on a small number of cases where we have the ability to rapidly strip out the autoantibody without using any immunosuppression. So all we're doing is taking out the autoantibody over the case of a week and then following up the patients for a year to see whether we have modulated the immune regulation. So we can analyze cells from these 2 studies. We're attempting to develop for the first time an assay that will pick up B cells that might have the receptor for PLA2R using a fluorescent derivative of our P28mer. And we're currently setting these studies up to do in the next few months. The other thing we're quite interested in is whether we can use this peptide as a peptide coating on an immunoabsorption column instead of just taking out total IgG on protein G type columns that are currently used in standard practice. But what progress have we made with Tcell epitopes? You'll be aware that, as I said earlier, this is a very genetically restricted disease where DQA1, we think it's DQA1, is the presenting class 2. And so the peptides that actually will interact with class 2 are quite important. And I think this is where the availability of assays like the ProPresent assay that ProImmune offer is quite important, because we've done some early experiments using 3 donors that are tissue typed, and this is the molecular typing that is the high-risk version, DQA1 0501, DQB1 0201. This genetic type is associated with the highest level of anti-PLA2R production in vivo. And so using 1 milligram of recombinant PLA2R per donor and going through the antigen presentation experiment as outlined in the cartoon here, and looking at the peptides that are contained in the class 2 receptors and getting the sequence from mass spec experiments, we have 5 potential candidates from those 2 donors. You can obviously try and predict in silico, and a standard in silico prediction algorithm would suggest that those sequences highlighted in red might be potential antigens, but our 5 candidates are not present in that list. And what we hope to do is to exploit this knowledge, again, using these cells that we've spent 3 years collecting, to see whether any of these 5 15-mers that we've got, whether we can use the class 2 tetramer approach, or whether we simply use standard ELISpot for looking at cytokine release when we stimulate patient T cells with these peptides, whether we can show that any of those candidates are potential peptides for taking forward as a T cell peptide. And we can then go back into our cell set here and see whether the frequency of these specific T cells is modulated over time. Is it very high when the disease is very active? Do they disappear when patients go into remission? Do they come back when patients relapse? But in addition to that shared sequence motif between the 2 autoantigens, you'll be familiar with the theories of autoimmunity and mimicry with infection. And just in this cartoon here, this is our PLA2R peptide, just shown in freehand here. But this half of it is shared by quite a few infectious agents, and I've just picked out one here that we're quite interested in. There are 100 Clostridia species. Some of them possess this cell surface carbopeptidase, others don't, but the homology is retained. And we've shown that anti-PLA2R autoantibodies can actually recognize that sequence in ELISA-type experiments. So there is definitely the potential for infection, be it this agent or others, to stimulate B cells that will have a receptor that can partially interact with the autoantigen PLA2R, and maybe under certain circumstances, those B cells could actually present PLA2R protein and initiate an autoimmune response. The question is, how can you study that? You need better reagents, and I've tried to outline to you that how we might take this forward by monitoring, looking for specific PLA2R specific B cells, monitoring for specific PLA2R specific T cells, and we're also interested in looking for the presence of soluble PLA2R, which I think is probably involved in maintaining and losing tolerance. So in summary, I've taken you through a fairly quick update of the state of our understanding of autoimmunity in membranous nephropathy. It's early days, we've made some progress, and I think the challenge now is to use this knowledge and actually, in addition to using the autoantibody as a biomarker, to be able to monitor T and B specific cells in different stages of the disease. And that's the team who've been involved in doing it. Thank you very much.